Good morning, everyone. Today we have a special webinar with a special speaker at a special time, Thomas Yeo, who has been our colleagues and friends for many, many years. We are very glad that he can virtually come back and give us presentation. So Thomas Yeo is an assistant professor at the National University of Singapore. His lab develops machine learning algorithms for large-scale brain imaging data sets with the goal of discovering principles of brain network organizations, how brain networks are organized to support cognition, and how brain networks are disrupted in mental disorders. Thomas is an editor at Neuroimage and eLife. He's a recipient of the OHBM Early Career Investigator Award. So without further introduction, let's listen to Thomas' talk. And before Thomas started, I just want to remind the audience that please use the QA box to write your questions rather than using the chat. Okay, Thomas.
Hi, everyone. Thanks for the invitation and thanks for the introduction. I was actually supposed to visit before the Montreal OHBM, but as you all know, because of the pandemic, no one can travel. So I'm still happy to do this virtually, but I really hope to come back and see everyone at a different time. Maybe in like a few years, who knows, right? Okay, anyway, so as Hui was explaining, my lab develops and applies machine learning algorithms to generate discoveries from large-scale brain imaging data. Today, I'll talk about our work on using generative models to explore resting state parcellations and brain dynamics. So I'm sure everyone knows that the brain is highly active even during the resting state. Furthermore, spontaneous fluctuation during the resting state can be used to interrogate brain network organization. For example, if we consider a superior parietal seed region and correlate its time course across the cortex, we get a network of brain regions shown here. This is called function connectivity, and this network of regions is known as dorsal attention network. So if we let the seed regions move around, we see that the correlation pattern changes a lot, suggesting that adjacent brain regions can have very different connectivity patterns. This has motivated many studies to extract brain networks or parcellations from resting state fMRI, which can in turn be used to provide insights into human brain organization. There's of course also a lot of interest in using resting state function connectivity to study neurodegenerative and psychiatric disorders. And we can also use resting state fMRI to study individual differences. For example, we can use function connectivity to predict fluid intelligence in individual participants. And the recent paper, actually, it was just published yesterday, showed that wearing a cast leads to a dramatic drop in motor network function connectivity, which then recovered quickly after removing the cast. So to summarize, functional connectivity is a powerful tool for exploring human brain network organization, mental disorders, and individual differences. And in this talk, I will be using, I'll be focusing on our work on using generative models to explore resting fMRI data. But first, what is a generative model? A generative model is a model that can be used to generate observed data. This sounds a bit circular, but I'm sure everyone knows of the most famous generative model, which is Newton's 3 laws of motion. If we know the initial states of the balls and the force applied by the pool table Q, right, then Newton's 3 laws allow us to predict how the balls move. This is an example of a physical generative model. But in neuroscience, we can have a whole range of generative models, right? At one end are physically realistic models, such as spiking neurons and so on. These models can be highly detailed. For example, a single cortical column can be modeled using thousands of parameters. At the other end are more abstract statistical generative models. For example, probabilistic ICA is a statistical generative model. The reason why I like generative models is that they usually make explicit claims about processes underlying the data. Therefore, the parameters are usually more interpretable and can potentially be used to derive insights into the brain. For example, there's a lot of work conceptualizing hierarchical relationships among behavior, neurocomputations, and neuroanatomy, or task, processes, and brain activation. Similarly, a hierarchy has been stretched out for mental disorders, which is kind of a major motivation for the RDoC initiative. So in this illustration, you have a brain system or brain circuit, right? And it supports multiple related cognitive processes. Deficits in these processes leads to symptoms, right? Some symptoms might be criteria for disorder A, some might be criteria for disorder B, right? And some might overlap across disorders. These are conceptual models that I'm showing here. They're not really mathematical models, which we can fit to data. So a few years ago, we formulated a generative model that captures the notion that behavioral tasks share multiple cognitive processes, which in turn activate overlapping brain regions. So the cognitive processes are not directly observed. However, we can estimate them by fitting the model to brain activation maps from 10,000 experiments across 83 tasks. This allows to infer the probability that a task would recruit a cognitive process and the probability that the process will activate a brain region. The probabilities allow us to infer interesting properties about brain organization. For example, my collaborator Maxwell, he shows that complex tasks, which he defines as recruiting more cognitive processes, these complex tasks result in greater activation at connector hubs. But if he looks at local hubs, then activations at local hubs remain constant regardless of task complexity. So this suggests that connector hubs might integrate information across specialized brain networks, supporting our ability to execute diverse tasks. Similarly, there's been work suggesting significant heterogeneity in late onset Alzheimer's disease. My student Xuming formulated a generative model, which encodes the idea that Alzheimer's patients share multiple latent factors associated with distinct patterns of brain atrophy. By fitting the model to voxel-based morphometry data, Xuming found 3 factors associated with distinct patterns of brain atrophy: cortical, subcortical, and temporal atrophy. So each dot here represents a participant. The corner of the triangle represents a pure factor or subtype. So this red dot here is actually almost like a pure cortical subtype, which is actually kind of interesting. So this means that this patient has predominantly cortical atrophy, which is interesting given that Alzheimer's disease is typically associated with medial temporal atrophy. However, most patients are in the middle of the triangles, expressing multiple atrophy factors, which might relate to co-occurring disease processes. So this type of hierarchical Bayesian modeling is quite flexible. Here we apply to structural voxel-based morphometry data because it is sensitive to Alzheimer's disease. Another student, she extended the model to deal with functional connectivity data, which allow us to look at autism spectrum disorder. She found 3 factors with distinct patterns of abnormal functional connectivity. And like before, each participant can express multiple factors, which might relate to co-occurring disease processes. Because of limited time, this is actually not the focus of my talk today. Instead, I'll focus more on brain parcellations and dynamics. So the remainder of the talk is divided into 3 parts. In the first part, I'll talk about statistical generative models for brain parcellation. Then I'll talk about statistical generative models for brain dynamics. And finally, I'll talk about biophysical generative models of brain dynamics. So let's move on to the first part. This is work done by my postdoc, Ruby. As I was saying earlier, resting state fMRI can be used to map large-scale networks in the brain. This is the 17 network parcellation from my 2011 paper. These networks are obtained by averaging data across 1,000 subjects. The problem is that group level networks do not capture individual specific network topography. So today I'll show you Ruby's approach for estimating individual specific networks. We also show that individual specific network topography can be used to predict behavior in individual subjects. Because we're talking about individual specific networks, obviously all algorithms try to take into account inter-subject variability, right? Because these algorithms need to estimate the networks in individual subjects. So you have to handle, the algorithms have to handle inter-individual differences. But most approaches do not account for intra-subject variability. This is important because inter-subject and intra-subject function connectivity variability can be very different across brain regions, right? So for example, we know that the motor cortex exhibit high intra-subject, but low inter-subject variability, right? So this means that if we see that the motor cortex function connectivity are very different across two subjects, then if we're not careful, we might mistake this difference for inter-subject differences rather than intra-subject variability. So Ruby developed a multi-session hierarchical Bayesian model that takes into account both inter-subject and intra-subject variability. And of course, for this to be useful, the algorithm also works with one session of data. This is the model. I won't go into it in details, but basically the top level models inter-subject variability. The next level models inter-session variability within a subject. And the bottom level measures inter-region variability within the network. We can learn all the priors shown in red from multi-session fMRI data. And given these priors, we can estimate the parcellation of a new subject with only one fMRI session. So Ruby trained her model on multi-session data from the Genomic Superstruct project, which is, of course, data contributed by many investigators in the Boston area, including many folks from the Martinos Center. She applied the model to the HNU dataset. The HNU dataset contains 10 sessions for each subject. So this allows us to test how much the algorithms improve as we use 1, 2, 3, 4, or 5 sessions of data to parcellate a subject. We use functional homogeneity as a metric of parcellation quality. So we define homogeneity as the correlations between time series within each network. Higher homogeneity means that the networks are more coherent, so the higher the better. So we apply the homogeneity metric to the leave-out sessions. So these are data we did not use to derive the parcellations. The gray bars correspond to Wang 2015 and Gordon 2017. The white bars are Ruby's approach. So the white bars are higher. So we have the best homogeneity. As the number of sessions increases, homogeneity improves across all algorithms, but the improvement seems to taper off around 4 or 5 sessions of data. So here each session is about 10 minutes, so around 40 minutes of data. So furthermore, using one fMRI sessions, Ruby's approach is basically equivalent to Wen and Gordon using 5 fMRI sessions. So the individual parcellations, if we look at the spatial maps, they look very replicable across sessions, right? So here are 4 subjects. Each column is 1 subject. The first row is day 1 and the second row is day 2. We see that there are fine-grained network features that are highly replicable across sessions, as shown by the yellow and the yellow and the blue arrows. So this topography is fine-grained and it's actually quite replicable within a subject, right? And there are big differences across subjects. So we then wonder whether these features are meaningful, right? And we did that by showing that we can use these features to predict behavior. Many papers have demonstrated that the size and topography of individual specific networks are different across individuals. But there's actually very little work indicating if, let's say, network topography or size are actually behaviorally relevant. Here we consider 58 behavioral measures from the Human Connectome Project. We use kernel regression to predict behavior. So what kernel regression does is that basically we predict the behavior of a test subject based on the weighted average of the training subject's behavior. So basically a training subject is weighted more if the training subject's parcellation is more similar to the test subject. So this allows us to test different hypotheses about what network features are important for prediction. So for example, we can define similarity based on spatial topography. right? So two parcellations are similar if they have higher spatial overlap, right? So basically, if my brain's default network overlaps a lot with Bruce Fishel's default network, right? Then we will assume that we have very similar behavior. We can also define similarity in a different way. In this case, for example, network size. So for example, is my default network is really big and Bruce's default network is also very big, right? Then we can kind of assume that the model will kind of assume that we have similar behavior and it will predict accordingly. So here's the result of the behavior prediction using network topography, right? So network topography is measuring how well two networks are overlapping, right? So some measures have high accuracies and some have low. So because we're averaging across 58 measures, so the result is kind of low, around 0.13. As a baseline, the red line shows the accuracy of the HCP megatrol. So the HCP megatrol uses functional connectivity strength for behavior prediction. So we can see that using network topography for prediction is not worse than using functional connectivity strength. Network topography estimated by Ruby's approach gives better prediction than Wang and Gordon. Finally, network topography is more useful for prediction than network size. I've just shown you kind of an approach for estimating individual specific networks. But we know from system neuroscience that the cerebral cortex comprises roughly 300 to 400 cortical areas, kind of with distinct function, architectonics, connectivity, and topography. So each large-scale network contains multiple cortical areas. So Ruby extended her previous model to estimate individual specific area level parcellations. I'll basically use the same, we'll basically use, well, not I didn't do any work. Ruby basically used the same multi-session hierarchical Bayesian model, right? But this time what she did was that she trains the model by initializing with the Schaefer group level parcellation. So we've previously shown that the Schaefer parcellation is highly homogeneous across multiple resting and task data set with different pre-processing, including global signal regression, ICFX, COMCORE, and so on. The parcellations also aligns well with traditional area boundary. But for example, shown here, but you can see that what happens is that the parcels, which are shown in blue, are actually fractionating these areas. So for example, if you look at area 3, you can see that the hand and the face region, sorry, the face and the hand regions, right, split into different parcels. So this is why I put a quotation mark around aerial, right, because the parcels do not correspond to traditional cortical areas. You can think of this as a weakness or a strength. I think of this as a strength because it's my parcellation, right? Because we are modeling a behavioral. So, for example, if you are modeling a behavioral task with button presses, it is probably useful to distinguish the hand region from the face region. We have some additional gradient-based priors to encourage strong transitions in functional connectivity profiles at the parcel boundaries. Like before, the area topography are highly replicable across sessions. Here are two midnight scanning club subjects. The top row corresponds to the first 5 sessions, and the bottom row corresponds to the last 5 sessions, right? Again, we can see these fine-grained aerial features that are different across the 2 subjects, but very replicable within a subject. And again, we wanted to see if these parcellations can be used for behavior prediction. Ruby again consider 58 behavior measures from the Human Connectome project. Here's the prediction accuracy from the previous individual specific network topography. The accuracy is better if we use the Schaefer group level parcellation to compute these 400 by 400 functional connectivity matrices, right, and use them for predicting behavior. And if we use the individual specific area level parcellations, we get a further improvement. Our prediction accuracy is also better than a recent individual parcellation approach. So the comparisons here are using hard parcellations in the sense that each brain location is assigned to a single parcel. we have performed further comparisons with other forms of data representation. So the setup is the same as before, 58 behavioral measures, 24 cross-validation repeated 100 times. Here are the results for the Schaefer group level parcellation and Ruby's individual specific area level parcellations. Well, you might not notice, but the numbers are actually a bit higher than before because we're using partial correlations instead of Pearson's correlation. And as other groups have shown, partial correlations seems to be better for predicting behavioral traits. The blue results are showing the ICA dual regression and Profumo. Everyone knows what's ICA, I'm sure. We tried different number of ICA components, 50, 100, 200, and 300. In this particular experiment, 50 works the best, so that's why I'm just showing you 50, right? Profumo is a relatively new approach, also from the FSL group. And finally, local gradient. Local gradient is the approach pioneered by Steve Peterson's group. Principal gradient is the recent approach developed by Daniel Margulies, where he applies nonlinear diffusion embedding to functional connectivity data to derive these principal gradients. And as we can see, generally speaking, the parcellation seems to do better than the gradients, and the individual specific area level parcellation seems to do the best. We have repeated this with linear rich regression, and the conclusions are similar. We're now actually trying to replicate this in another data set. To summarize, I've shown you the multi-session hierarchical Bayesian model for individual specific cortical parcellation. The approach accounts for both inter-subject and intra-subject variability. Compared with other approaches, Ruby's parcellations require less data for comparable performance. When we use the same amount of data, Ruby parcellations are more homogeneous in out-of-sample resting and task fMRI data. And we also show that individual specific parcellations can improve behavioral prediction. The model, the multi-session hierarchical Bayesian model, it needs multi-session data for training, but once it's been trained, it can be applied to single fMRI session, single session fMRI data. So at this point, I am actually at the end of part 1. Does anyone have questions?
Yes,
or?
We have Lila Soleil that has a question.
Hi, Lila, how are you?
Hi, Thomas. Very quick question. You mentioned the Lee 2019 paper when you compared Ruby's results.
Yeah.
By the, can you just quickly summarize how those two approaches are different?
The Lee 2019 is kind of this from Heshang's group. They basically do like a template kind of matching approach. Yeah, to derive the individual parcellation.
Okay. All right. Thank you.
Should I continue? Okay. So I will now move on to the second topic where we explore a simple generative models for fMRI dynamics. This work was done by my postdoc, Raphael. So in the previous part of the talk, basically functional connectivity was computed by correlating the entire fMRI time series. So this assumes that the underlying functional connectivity does not change over time. Therefore, what many studies have done is to compute functional connectivity over short time windows. So for example, here's a functional connectivity matrix computed using fMRI data during this short time window shown here. We can slide the window, right? We can slide the window and then we can compute another functional connectivity matrix, okay? And then we can keep doing that. And so we get a whole time series of functional connectivity matrices, right? People call this time varying functional connectivity or dynamic functional connectivity, right? So what do we do with so many matrices, right? Because now instead of a time series, multiple time series, we now get a time series of matrices. There are many ways to look at these matrices. For example, we can visualize this by computing correlations between all pairs of these matrices, right? So if we compare, if there are, if we compute, for every pair of matrices, we compute a correlation, right? We end up with another correlation matrix shown here, okay? So essentially this is a T by T matrix where T is the number of sliding window matrices, okay? We call this the functional connectivity dynamics matrix. Well, actually, we didn't call this. Previous people call this the functional connectivity dynamics matrix. Okay. The diagonals of the functional connectivity dynamics matrix is high because of the autocorrelation in the fMRI data and the overlapping sliding windows. What is very intriguing are these off-diagonal blocks of basically kind of recurring functional connectivity patterns. A major question is whether these recurring connectivity patterns are evidence of discrete brain states. To answer these questions, we can fit different generative models to the fMRI data and check which models can reproduce the observed dynamics. So a simple generative model is the first order autoregressive model. So the idea is that the fMRI data at time point t+1 is a linear combination of the fMRI data at time point t, right, plus some noise. So previous work has fitted bivariate autoregressive models, right? This means that each pair of brain regions is fitted independently, right? And you can see that if you do that, then we get very flat functional connectivity dynamics, right? Where there's no recurring connectivity patterns. On the other hand, if we fit hidden Markov models to the data, we can generate recurring connectivity patterns, right? Because hidden Markov models explicitly model brain states, right? We can interpret this as evidence for discrete brain states. But it is worth noting that the hidden Markov models tend to generate connectivity dynamics with very sharp transitions, right? That you do not see in real data. Finally, what Raphael did was that he fitted a multivariate autoregressive model based to all brain regions simultaneously, right? So instead of fitting every pair of regions separately, he fit all of them together. And this actually generates quite realistic functional connectivity dynamics. So this is quite interesting because the autoregressive model does not assume any discrete brain states. And I'm not saying that there is no discrete brain states, right? I'm just saying that we can actually observe this kind of dynamics, right? Without the need for discrete brain states. In a follow-up study, Raphael used the variance component model to show that the multivariate autoregressive model can explain behavioral data better than static functional connectivity. He then sorted the 58 behavioral measures, right? So on the left, static functional connectivity is better, and on the right, multivariate autoregressive model is better. Raphael then colored self-reported measures orange and task performance measures green, right? And we see that the orange measures seems to be clustered on the left, and the green measures are clustered on the right. And indeed, if we only consider self-reported measures, there's basically no difference between static function connectivity and the autoregressive model. But for task performance measures, the autoregressive models is better than static function connectivity. This section is quite short. To summarize, static function connectivity, right, does not account for temporal dynamics. With sliding window correlations, we can visualize compelling functional connectivity dynamics. And these dynamics can be reproduced by multivariate autoregressive models without assuming the presence of discrete brain states. Autoregressive models can explain task performance better than static functional connectivity. However, this is not the case for self-reported measures. Overall, Raphael's result shows that resting brain dynamics at different time scales capture distinct aspects of human behavior. This is a short section. I'm done with it. Do people have any questions or anything?
No, you can go ahead, Thomas.
Sure, why not? Okay.
Well, sorry, actually.
Bruce Fisher, let me. Bruce, you want to ask?
Hey, Bruce.
Hi, Thomas.
How's it going?
Pretty good. So my question is, you seem to be suggesting that there's kind of a static resting state network that's going on all the time that gets recruited by the brain for doing some task. Is that the case?
Oh no, sorry. I was not being clear. This prediction is actually for traits. In all of these cases, I'm using it to predict traits. So these are behavior measures that were measured outside the scanner. Sorry, I was not being clear. There are, of course, other people who have tried to relate the time-varying dynamics to ongoing cognition, yeah, which is of course much harder to do. Yeah.
Okay, thank you.
Yes.
Okay,
sorry.
I guess there's no more question.
Okay. So even though the auto-regressive model explained the fMRI dynamics very well, but it is a statistical generative model. So in some sense, it can provide certain insights into the brain, but deriving mechanistic insights is tricky. So for the last part of my talk, I'm going to talk about inverting neural mass model. This work was done by my postdoc Peng Wang and my student Xiaolu. So as we all know, the human brain is a complex system spanning from micro scale to the macro scale. But our knowledge about how these scales interact is extremely sparse. A promising approach to bridge across these spatial temporal scales is, of course, through biophysical modeling. As I was saying before, generative models exist on a spectrum, right? On one end are biophysical models, whereas on the other end are more abstract statistical models. So today I'll focus on neural mass models. They are closer to the biophysical end of the spectrum, but they are not super biologically realistic, right? But even then, a lot of people have shown that they can be useful for providing mechanistic insights into the dynamic resting brain. Before I tell you about neural mass model, I kind of wanted to first describe kind of the framework of how people explore neural mass models, at least in fMRI data, right? So people, suppose we have a structural connectivity matrix. This matrix can be from diffusion MRI data. It can also be from invasive track tracing studies in animals. We can apply a neurodynamical model to generate simulated neural signal. We can then apply a hemodynamic model to simulate fMRI time courses and the corresponding functional connectivity matrix, right? So this simulation does not include task inputs. So we are generating simulated resting state function connectivity. And what people then do is that they compare empirical or observed real functional connectivity data with the simulated function connectivity data, right? And the assumption here is that if the simulation and the real data are in good agreement, then this implies that the neurodynamical model is a good model of brain dynamics and can therefore be used to provide insights into the resting brain. Most papers just use the balloon model, which is what we'll use here. Today we'll focus on the neurodynamic model. So there are many possible models. In fact, that's what I understand what computational neuroscientists do. They come up with different biophysical models every day, apparently, right? So we will just focus on one model, the dynamic mean field model from Gustavo Deckel. So the model captures the average dynamical behavior of cortical regions at the level of neuronal populations, right? So in comparison to more detailed spiking neuronal networks coupled by AMPA, NMDA, and GABA receptors. So this mean field model is much simpler, but it's able to generate large-scale brain dynamics with relatively low parametric complexity. So in the mean field model, each cortical ROIs is represented by a system of nonlinear ODEs, ordinary differential equations, coupled together by structural connectivity. The neurodynamics at each region is driven by 4 types of inputs. The first type is the intra-regional input and is parameterized by the recurrent connection strength W. The second type of input is the inputs from other brain regions, right? So the inter-regional input is parameterized by the structural connectivity matrix, scaled by this global constant G. The third input is an external input current I, potentially from subcortical relays. And the fourth input is neuronal noise, parameterized by the amplitude sigma. Gustavo and colleagues have shown that there's good agreement between simulated and empirical function connectivity, right, when the brain operates close to the edge of criticality. So one tricky situation is that the 4 parameters are manually set, right? And furthermore, they assume that, well, they assume that the recurrent connection strength W and the external input I are the same across all regions, right? But given that different cortical regions have distinct cellular properties, this is just not very biologically plausible, right? But this is understandable because they are manually tuning their hyperparameters, right? So if they allow different brain regions to have their own parameters, then manual tuning is impossible. So our goal then is to automatically estimate the parameters of the mean view model. Because we're automatically estimating the parameters, we allow the recurrent correction strength W and the external input I to vary across the brain. In total, we have about 138 parameters, and we estimate these parameters by maximizing the agreement between the empirical static function connectivity and the simulated static function connectivity. We use data from the Human Connectome project, and we split the data into training and test sets. We estimate the parameters using the training set, and we evaluate the fit of the test set. So after we estimate the 138 parameters of the relaxed mean view model, the agreement between the empirical function connectivity and the, hold on, which one is it? Simulated function connectivity, right, is about 0.48. If we simply optimize the original mean view model, so this is the model whereby they assume that all the parameters are constant across the cortex, right? So then the agreement is only 0.27. And if we simply correlate the structural connectivity and the empirical function connectivity, right, the agreement is actually 0.3. So this means that actually the baseline agreement between, even if your model does nothing, right, because if you correlate structural connectivity and the empirical function connectivity, right, you get a correlation of 0.3. So essentially the original mean view model is basically not doing much. This is actually important control conditions, but many papers still do not report the correlations between structural connectivity and the empirical function connectivity. So we get an improvement, but you know what? Is there a spatial pattern to the parameters, or they're just kind of random? What is very cool is that they are not random, right? So on the left is the external input currents for each cortical regions. The purple color means that the input current is high. The light blue means the input current is low. So the black boundaries here, shown here, corresponds to the 7 network parcellations from my 2011 paper. The yellow arrows are pointing basically to like early sensory motor regions, right? Visual, motor, auditory cortex, right? So we conclude that sensory motor regions have stronger external inputs, potentially from subcortical relays. The yellow arrows are pointing to the default network. You can see on the left, the yellow arrows are pointing to mostly light blue regions, light blue colors. So basically, the default network has weak external inputs. The recurrent connection strength also shows a similar patterns. So what is kind of cool is that without assuming the existence of a cortical hierarchy, the estimated model parameters reveal a large-scale cortical gradient. We compare actually our model estimates with Daniel Margulies' principal gradients, and we find that the recurrent connection strength and the external inputs are correlated with Daniel's principal gradients. Finally, we obtain cellular density estimates from Martin van der Heuvel. We find that the recurrent connection strength is correlated with neuronal cell density, but there's no relationship with the external input currents. And we repeat the analysis with the cell size estimates, and there's no correlation with recurrent connection strength or the external inputs. So in Peng's paper, we actually maximize the agreement between the static function connectivity in the simulated data and the real data. So we were wondering whether the relaxed mean view model generates realistic functional connectivity dynamics. This is the a repeated figure. This is the function connectivity dynamics matrix of a single subject. This is the function connectivity dynamics matrix where Raphael fitted a multivariate auto-regressive model, right? This is the function connectivity dynamics of the relaxed mean view model where we fitted the static function connectivity. And you can see that we get this very flat function connectivity dynamics with no recurring connectivity patterns. This is very sad because the mean field model is supposed to be more biologically realistic, right? And it's much more complicated than the auto-regressive model, right? But yet it generates much less realistic dynamics. And the important lesson here is that more complex, more realistic model might not actually explain your data better. So the issue here is that because we only fit it to static function connectivity, right? So we essentially overfitted to it. So what Xiaolu did was that he developed a new algorithm that allows us to fit to both static function connectivity and function connectivity dynamics. And we get much more realistic patterns dynamics shown here. And what is very cool is that the spatial topography of the mean field model still demonstrate a macro scale gradient. Another interesting thing is that even though we're fitting to both static function connectivity and function connectivity dynamics, right? We also improve the fit to static function connectivity, right? So this is the agreement between the static, the fit to the static function connectivity. This is in the test set, is 0.48, right? When we try to fit to static function connectivity. Here we're fitting to both static and dynamic function connectivity, right? And yet in the test set, the fit to static function connectivity is better. And probably the reason is that the mean field model is highly nonlinear. The space of the mean field model is very big. So in fact, by adding more things to optimize, we actually get a better fit overall. We get a better solution. The next thing we want to do is that now we have this, we want to dig kind of deeper into kind of the mechanisms of the function connectivity dynamics. This is the function connectivity dynamics of the simulated model. So just a reminder, there are time periods whereby the functional connectivity patterns are stable over a long period of time. As you can see by this block of high correlations, right? There are also time periods in which the functional connectivity patterns are stable only for a short period of time. So basically this red diagonal is just showing the autocorrelation data. So I do not know much about nonlinear dynamical systems. I'm sure there are audience members who actually know more than me, right? But Xiaolu tells me that these periods might correspond to the brain system being at this criticality zone, right? In which the brain exhibits multi-stability. So there might be this period during which the system is functioning in this low amplitude state, right? And in this state, the system is basically dominated by noise. So as a result, you have this, low coherence. And then there might be a period during which the system is functioning in this high amplitude states. In this state, there are this large oscillation in the data, right? And so you get this large coherent block. Now, if this is true, then in the low amplitude state, right, we will predict that there's small standard deviation, right, in the Bose signal. And in the high amplitude state, we would predict that there will be large standard deviation in the Bose signal. So let's see if this is true. So on the left is the simulated function connectivity dynamics matrix. On the right is the function connectivity dynamics matrix from a real subject. So we want to turn this 2D matrix into a 1D time course, right? So we simply average the rows of the matrix, right? So if we average the rows of this matrix, we get a time course shown here and the time course shown here, right? And we can see this sharp transitions in time course seems to correspond well to the switching in the function connectivity dynamics matrix. So essentially this time course is a good representation of this, okay? So at the bottom, each time course here, so at the bottom, I'm also plotting the time courses, right? Each time course represents a brain region, right? The time course, each time course corresponds to the standard deviation within the sliding window of the Bose signal. So we see that there are brain regions in which there's sharp transitions in the standard deviation time course, and they seem to track the switching in the functional connectivity dynamic matrix very well. So more specifically, basically during these periods of high signal amplitude, right, functional connectivity patterns are stable over a long period of time. On the other hand, during periods of low signal amplitude, functional connectivity patterns are unstable. So this suggests that our prediction is working out quite well. Basically, based on let me think, which one's the simulated? Based on the simulated and real data, it seems that the period of high coherence seems to correspond to big fluctuation in the signal. The period of low coherence seems to correspond to low fluctuation in the signal. So then we wanted to see if certain brain regions might be driving this switching dynamics, right? So we correlate each of this sliding window time course with the functional connectivity dynamics mean time course. And so we get a spatial map like this, right? So on the left is the simulated data, on the right is the real data. Purple color means that, purple color like these means that sharp transitions in the standard deviation time course seems to track switching in the functional connectivity dynamics. The correlations are actually not that high, which is quite surprising because if you see from the previous slide, it seems to match really well. And this is actually representative subjects. These are representative subjects. So, but anyway, so I'm not sure why the correlation is a bit weak, but anyway, seems visually looks quite well, but these are highly significant. We see that sensory motor regions seem to have high values, right? So this means that the standard deviation of sensory motor, time courses seems to track functional connectivity dynamics. And you can see that there's a strong agreement between the simulated spatial map and the empirical spatial map. Finally, Kevin Anderson from Avram Holmes Group recently published a paper where he shows essentially the gene expression map for somatostatin and parvalbumin, right? So blue means that there's an overexpression of somatostatin, and red means that there's an overexpression of parvalbumin. The gene expression maps, actually, if you look at this, it seems quite similar to the spatial maps I've shown you before. And what is pretty cool is that Kevin's paper and previous papers have shown that somatostatin interneurons are linked with low, hold on, did I screw up? Hold on. Oh yeah, sorry. So this is somatostatin neurons, right? Somatostatin neurons are linked with low neural signal amplitude, whereas the parvalbumin interneurons are linked with high neural signal amplitude. So this is consistent with our results showing that both signal amplitude changes in sensory motor regions seems to track functional connectivity dynamics. And indeed, the gene expression spatial maps and our spatial maps are strongly correlated. So to summarize, we have demonstrated that it is possible to automatically fit a neural mass model. This allows us to explore more biophysically plausible models where each region has its own micro scale properties. By using the relaxed mean field model, we improve the agreement between, with static function connectivity and functional connectivity dynamics. And what is very cool is that the estimated model parameters kind of review this large scale cortical gradient. We also found that the time varying amplitude of sensory motor regional fMRI time course seems to track the functional connectivity dynamics. And furthermore, it may be the case that parvalbumins might be more strongly expressed in sensory motor regions and has been linked with greater signal amplitude. And this kind of provide a potential mechanisms for the functional connectivity dynamics we observe. Overall, I'm very excited about this line of research. By using machine learning to invert biophysically plausible generative models, right? We're able to link macro scale cortical hierarchy with micro scale brain organization. So basically today I have discussed generative models of cortical parcellations in dynamics. We can use generative models for analyzing brain data, but we can also use generative models as models of the brain. An important lesson is that complex, more realistic models are not necessarily better, right? For example, a simple auto-regressive model can explain fMRI dynamics as well as the more complex, but more biologically realistic neural mass model. So all our code, parcellations, and models are available on GitHub. I'd like to thank all my funding sources. And of course, I also like to thank past and current members of my lab. I've highlighted those who have contributed to this particular presentation. And I'd like to thank all my collaborators.